Welcome back to Genetics in the Paddock with Emily. I'm your host, Emily Johnston, and I'll be taking you along on my journey to learn about all things genetics in extensive livestock. Last season, we explored the foundational aspects of genetics. We discussed everything from basic genetic principles to practical applications on farm. This season, we're kicking it up a notch. So with that, let's get into this month's episode. Welcome back to another episode of Genetics in the Paddock with Emily. Today I'm joined by Gus McFarlane, who is my guest speaker of the episode. And I'd like to say a big thank you, Gus, for sitting down to have a chat with me about some interesting topics we haven't actually really discussed on the podcast yet. So I'm really interested into diving into some of them.
Yeah, thanks for having me on. Thanks for the invite. Keen to have a chat.
Thanks, Gus. So I might get you to just start off with just introducing yourself. You can go as broad as you want, you can go as in detail as you want, but just tell me a little bit about you and some of the work that you're involved in.
Yeah, so I'm a research scientist at New South Wales DPI, and my work focuses on the application of gene editing tools and how they can improve agriculture, but also be applied for diagnostics. And before joining New South Wales DPI, I was developing gene technologies over in the UK. I did a PhD and a postdoc over there at the Roslin Institute, but I'm originally from Australia, so the weather and the sun drew me back home, and I'm working at DPI now, trying to continue my stream of research and the benefits I think that it can potentially offer the livestock industries.
Cool, thank you for that. I actually went to the UK last year, and there is a clear difference in the weather. Like, it was beautiful, but there is something about the Australian climate. It's so nice to be here, so I can see why you were probably drawn back as well. But it sounds like you must have had a really interesting experience over there at the Roslin Institute. I'm sure a lot of it has shaped what you're interested in today, or some of the work that you're also doing, which I'm excited to dive into. So I guess just to give a bit of context for the episode, like, why is there growing interest in gene editing? And I've heard a term floating around, CRISPR. So why is there an interest in that? And can you tell me a bit about CRISPR as well?
Yeah, sure. So I think a good place to start would be, as farmers and humans, we've been selecting traits in animals since we first domesticated them thousands of years ago. So we've been indirectly modifying the DNA of animals for centuries now. We did that first by eye and more recently through genomic and genetic selection techniques. And gene editing is just the next step in that process. It's a more precise method of genetic selection. And the concept of gene editing, and actually the tools to do it, aren't really new. We've had the capabilities to do it for almost 3 decades now. But recently, we've had CRISPR arrive. So up until the arrival of CRISPR, gene editing could really only be done in highly specialized labs that were set up specifically for gene editing. And CRISPR arrived in 2012, and it's been game-changing. It's a relatively low-cost tool. It's highly accurate, and it can be done in any just well-equipped DNA lab. So just a standard molecular laboratory lab. And that really puts gene editing within reach of most livestock breeders in the developed world. So that's why there's been a more rapid increase in CRISPR and CRISPR tools, because it's essentially democratized the application or the potential application of gene editing in livestock.
Okay, it sounds like a really interesting and new technology that's come about. I'm really interested in unwrapping some of that a little bit more. So what are some of the key opportunities in gene editing that you see that could be beneficial for Australian producers?
Yeah, good question. So I personally think the big win that gene editing can offer livestock industries is the ability to transfer a desirable trait or a couple of desirable traits from one breed to another. So we can do that through crossbreeding, but when we do it through crossbreeding, we get unwanted genetic carryover. So a good example of this would be introducing polled or hornlessness trait into high-performance dairy cattle like Holstein Friesian. You can do that through crossbreeding with a polled animal such as Angus, and therefore you don't have to physically dehorn calves when they're born to protect the safety of animals and farmers. But when you do that crossbreeding, do get a subsequent reduction in milk yield. So there's been many efforts to try and crossbreed and then back cross with Holstein Friesian to get back to those original desirable high milk production traits, but you never get quite back to the same levels. So with gene editing, we can precisely take the genetics underlying poledness in Angus. We know it's called the Celtic polled allele, and we can introduce it into the Holstein Friesian, which has already been done in the US. And then we get a high-performance dairy cow that's also polled, and it doesn't have that loss of milk production traits. And the same could also be done for full-blood Wagyu, where you could introduce poledness into full-blood Wagyu, and you don't get a loss in those highly valuable meat marbling characteristics. But this is just one example, but really gene editing has the potential to be applied anywhere that we see a trait that's beneficial, be it disease resilience, thermotolerance, welfare, carcass or fibre traits. Any of these traits, if we understand the genetics underlying those, there's a potential to use gene editing to transfer that specific trait from one breed to another without having that genetic carryover. So from my perspective, that's the key opportunity that gene editing can offer livestock industries.
Absolutely. It sounds very powerful, and I can see why a lot of people would potentially be quite interested in a technology like that with all of the benefits that you just described. So I guess when I've heard a lot of talk around like the gene editing space and all of that sort of stuff, GMOs or traditional GMOs are something that has always kind of filtered into conversation or have always been brought up at some point. So can we talk a little bit more about how gene editing like CRISPR is different from these types of GMOs that we always hear about?
Yeah, for sure. It's something I'm very passionate about, trying to educate the public about the difference between traditional GMOs and gene editing. So traditional GMOs involve inserting an entire gene, they're transgenic GMOs, an entire gene from one species to another instead of breed to breed. So a good example of this would be the Enviro pig. It was developed back in 1999 by a team in Canada. Had a great goal. It was to introduce the phytase gene, which was found in E. coli bacteria, into pigs so it was expressed in their saliva. So that gene can then allow the pig to digest phytate, so it gets more phosphorus from its food, so it's got better nutrition, but importantly it's producing less phosphorus in its manure, which we know phosphorus can impact the environment and waterways. And in order to do that, they had to insert a 46,000 letter piece of DNA that contained bacterial genes into the pig's genome. So it had a great goal, but understandably it never took off. People just don't want bacterial genes in their pigs. Gene editing is entirely different to that old style GMO, that first generation GMO. Gene editing, like CRISPR, we're typically making very minor changes to the DNA that's already present. So we're looking to replicate the DNA that we're already seeing in that species into another breed, so breed to breed, such as Angus to Holstein, that's staying within the cow. And we're often making very minor changes. So we're talking about like 1 to 10 letters in a 3 billion letter genome. And if I just put that into perspective, because sometimes it's difficult to perceive how small these changes are. But if I look at my bookshelf and I find the thickest book, my wife's a Harry Potter fan, it's the Deathly Hallows book. It's the last one in the Harry Potter series that has about 1 million letters in it, according to Google. So if I take 3,000 copies of that book and stack them up, this would be representative of the animal's genome. If I take one of those books out and change 1 letter or 10 letters in those books and put it back in the pile, that's the kind of changes that we're making in the animal's genome. And it still amazes me today that we now have the tools with CRISPR to do that microscopically with high precision inside a cell. So that's the difference between gene editing and traditional GMOs. We're making very small changes to the DNA that's already present within that species compared to introducing novel genes into that species.
Okay, so there's a very clear difference from what I can understand between the two of them, and I really love that analogy. I think it makes a lot of sense for people who like to visualize concepts like myself. So it seems like quite a minimal change, but quite a large impact for a lot of the breeds and species that are currently being worked on in the gene editing space. And as we talk about CRISPR, I'm trying to imagine exactly how it works when you keep talking about changing a couple of letters. So I know it's probably something that's a lot easier to show, particularly with a visual, but can you kind of try and explain a little bit about how CRISPR works in this sort of context in some simple terms, if that's possible at all?
Yeah, I'll give it a shot. You're right, it does help with some visual representation, but if we think of CRISPR like a highly precise pair of scissors, like a molecular scissors, microscopic, and we can introduce those into a cell. In this case, it would be an embryo because we're looking to make gene-edited animals. And CRISPR basically has a GPS that will guide it to the exact site in the genome that we wish to edit. that GPS is a guide RNA, and it has to bind with a sequence of DNA. But once, anyway, the CRISPR enzyme's delivered to that spot, it works like a pair of molecular scissors to make a break in the DNA. And once it makes the break, we've also provided into the cell a repair template. And that's a small piece of DNA that contains the changes that we want to add into that break site. And the cell then uses that repair template to repair the break, and in doing so copies in the intended changes into that site. And those new changes in the DNA will then code for the trait that we wish to introduce. So that's kind of how it works at a molecular level. That's a simplified version. I hope it makes sense.
Yeah, no, that was perfect. That definitely makes sense. And it sounds like it's a very precise tool, which is really, really cool to hear about. I'm interested as well, because we just spoke about how it works at the molecular level, but what would actually be involved in the process of gene editing livestock embryos?
Yeah, so gene editing can be applied anywhere that IVF is currently being applied, and we know IVF programs becoming more and more used in livestock industries. That would work typically by collecting oocytes or eggs from a donor female or females, and they're in vitro fertilized. And shortly after the fertilization, we then do the CRISPR part in the process, which would be taking those fertilized eggs and putting them in a solution that contains our CRISPR tools, and they go into an electroporator, which is a small portable device. It fit inside a carry-on suitcase. It's quite small, and a pulse of electric current is applied across those embryos in the CRISPR solution, and that creates small temporary pores on the cell's membrane that allows the reagents to go inside the cell, where they do that molecular work inside the cell, but the pores seal up once the current has been removed. Then those embryos are then back in this exactly the same IVF system. They're cultured up to blastocyst stage. We can then take a biopsy of the embryo and sequence it to confirm that it's gene edited and that everything's correct. Those correctly edited embryos would then be transferred into recipient females, and they would develop into genetically superior animals, or animals carrying the trait that we've introduced into them. So that's how it works on a macro level or an on-farm setting.
Yeah, thanks for sharing that, Gus, because I think sometimes when we talk about science like this, it's almost like, oh, how would that work in the real-life application? But it's always nice to hear about how this could actually work on a farm level, and it seems like quite an achievable thing to do as well. So I guess at the start of this episode, we did touch a little bit on some of the current examples of gene editing that are currently being used or trialled in livestock. But are there any others that you can kind of think of, or particularly what traits are being targeted, specifically those that are relevant to extensive livestock systems, but also just interested to hear about what's happening in general? And I guess I'm good at asking really long questions as well, but to tackle on the back of that, are there any international examples that you can think of that show us what might be possible here in the next couple of years?
Yeah, so all the examples I could describe are international examples. So we don't have examples in Australia yet. But the first one that jumps to mind, and it's not an extensive system application, would be pigs that are genetically resistant to the PRRS virus, which is porcine reproductive and respiratory syndrome virus. The reason it jumps to mind is because at the end of April, it just got approved by the FDA to go into commercial production in the US. So there's now pigs that have genetic resistance to this virus. We don't actually have the virus in Australia, but it's devastating to European, American, and Chinese pig industries. In the US alone, it costs the industry $1.2 billion a year. But a company called Genus has been establishing a commercial herd for some time, and then got approval to go into the food system. So that's one example. I guess from an extensive farming perspective, we've got in the US, and also continuing down that disease resilience path, is cattle that have genetic resilience to BVDV virus, which is bovine viral diarrhea virus. Bit of a mouthful. But we have that here in Australia, and it's actually a significant economic and welfare concern around the globe. So USDA, a team at USDA, led that project. So there's cattle with resilience to that virus. From more of a reproductive standpoint, one that certainly would have benefits in Australia is slick coat phenotype in cattle. It was originally identified in Senepal and tropical breeds of cattle, but a company in America called Excelligent has introduced that slick coat phenotype into Angus. So these Angus then have better thermotolerance, they've got better tick resilience too, and they also don't get the same milk depression during hotter months, so they get better mothering ability. Closer to home, in New Zealand, they've actually introduced the diluter gene, which is a lighter coloured coat colour into Holstein Friesian cattle. So therefore, if they've got a lighter coloured coat, they absorb less heat from the sun. But the same could also be done for Black Angus or Black Wagyu as well, to get a coat colour similar to that of a Murray Grey. So they absorb less heat from the sun, basically better thermotolerance. In smaller ruminants, we've got Cashmere goats in China. They were gene edited to have a longer fibre length, and from a production setting too, we've got sheep, cattle, pigs, goats that have all been edited with differing myostatin gene edits. So myostatin is the gene that controls double muscling phenotype, so they get increased muscularity, so better carcass yields. They are all the examples that spring to mind for land-based agriculture, but you may or may not be surprised to know that there's many more examples actually in the aquaculture systems, and this is just typically because they have faster generation intervals and they produce many more eggs.
Yeah, that makes sense. Thanks so much for sharing that, Gus. It's interesting to hear just about the scope of what's going on in a range of different species, and it sounds like there's a lot of different things that can be targeted, from production and direct impact on animal welfare, which is always a nice benefit as well. So we spoke a lot about the international examples, but how close are we to seeing gene-edited animals in Australian paddocks?
So there's none in Australian paddocks as yet, but my perspective is we're not far off. We've got the science, that's ready. We've also got the tools and capabilities to both develop and validate and test imported genetics in Australia to ensure they're up to standard. The main hold-up is really the regulatory clarity around the space. So it's quickly becoming clearer in Australia and around the globe, but at the moment we need that regulatory clarity in order to get broader industry support for the potential that gene editing can offer the industry. So yeah, none at the moment, but hopefully or potentially in the near future.
Yeah, that makes sense. I was just about to ask actually about, I'm guessing gene editing would be heavily regulated in Australia, but who kind of oversees this space?
Yeah, we do have quite strong regulations in Australia. There's two main bodies that regulate the application of gene editing in livestock in Australia. The first is OGTR, which is the Office of Gene Technology Regulator, and the second would be FSANZ, which is Food Standards Code Australia New Zealand. So OGTR looks at animals that have been developed with gene technologies and how they could potentially impact human health or also the environment, but they don't look at the foods that are derived from those animals. So OGTR has already taken significant steps to reduce the barriers for gene editing, and it opens up many more research avenues. They've even deregulated one form of gene editing, so it's no longer under their GMO classification, where FSANZ, part of their remit, looks at regulating foods that derive from gene-edited animals. So that's important in order to commercialise food products from animals. And FSANZ has been looking to update their Food Standards Code in relation to gene technology since 2020. So what's that, almost 5 years now? Because this code was originally put together for transgenic GMOs, those ones that I described earlier. And the code hasn't really kept pace with the developments that we've seen in gene editing technology. But FSANZ has already undergone 2 rounds of public consultation on how they hope to update the code and their definitions around what's possible within the food system. And if we look at their current revised definition, it would allow the movement of DNA from breed to breed, because you're not actually adding any novel DNA to that species. So that would significantly streamline the application of gene editing in Australia. So really hopeful that we'll have an outcome from FSANZ in the next 12 months. So it will kind of give us a bit more clarity on what we can do and what can't be done. And I'd say it's a pretty exciting time to be involved with the technology, because we're seeing things happen in many places around the world that can benefit livestock industries and the environment.
Yeah, cool. I'm really interested to see what actually comes out of that. I think it'll be very, very interesting, yeah, especially as things continue to change and more science continues to become more available. And, there's obviously a need for some of these gene edits, or a desire for some of them as well, which is really interesting to hear. So it'd be great to see what comes out of that. So in this episode, we've spoken a lot about some of the major benefits for gene editing, but I'm interested to see, is there any sort of risk or limitation with gene editing? Because I have heard a bit about off-target effects, or are there any sort of unintended consequences that could arise? Let's talk a little bit about that.
Yeah, sure. It's always important to address the potential risks or limitations of the technology. And I guess the first ones that come to mind for me are around the technicalities of it, off-target effects, as you mentioned there. First generation, although it was very rare, there was potential for off-target gene editing. But we're fortunate there's been a lot of work around the tools, CRISPR tools for gene editing, particularly in the human space, where they're looking to use gene editing to address genetic diseases in humans. So obviously you want highly precise tools for that. And we're able to piggyback off those innovations to now have highly precise tools that very, very rarely have off-target effects, extremely low frequency. But an extra safeguard on top of that is that we always whole genome sequence any gene-edited animals that have been developed, and that ensures that there is an extra layer of validation that there's no off-target effects. From a technical standpoint, being a scientist, another one would be the potential for mosaicism. So mosaicism, pretty fancy sounding word, but it's when an animal has gene editing not in all of its cells, just some of its cells. And this is a result of gene editing occurring not when it's just a one-cell embryo, but when that embryo's gotten to multiple cells and editing's happened in some of those cells, but not all of them in the embryo. So it develops, those cells grow, and some of them, they propagate, some are edited and some are not. But we're fortunate that there's been lots of work done around this. We now have better protocols and the timing of when we should introduce the CRISPR reagents. And again, we have better tools now. So tools that are instantly active inside the embryo, they don't have a delay before they start their gene editing work. So from a technical side, they're the two I see. Again, I think from a regulatory standpoint too, that's a limitation with the technology where it currently is. So we want to ensure that Australian producers have the access to the best genetics in the world for their environment, but we also want to ensure that they maintain widespread market access. And different countries around the world are regulating gene-edited foods in different ways. And in some nations like Argentina, Japan, US and Canada, they've already got gene-edited foods and GMO animals or foods from GMO animals on their supermarket shelves. So they're being consumed in those countries. The UK has also updated its regulations very recently, but many countries, including Australia, are still undergoing review processes of how they're going to regulate gene-edited food products. So in time, I think we'll see an alignment of gene editing regulations around the world in countries, but as it stands, any edits that we're looking to make, we need to ensure that we maintain widespread market access for our producers. So that would be the limitations as it currently stands, I think, for the technology.
Yeah, absolutely. And interesting that you mentioned market access, because I imagine that's a pretty big thing for a lot of people. But interesting to hear that a lot of other countries already have gene-edited foods on their shelves. So it sounds like maybe Australia might not be too far behind them. And a lot of, just circling back just a little bit to some of the other episodes that we've spoken about on the podcast, but on the podcast we speak a lot about tools like EBVs and selection indexes and all of those sort of traditional genetic selection tools. How do you see gene editing working alongside these sort of tools, or do you feel like they might replace them?
Certainly not, no. EBVs and indexes are here to stay. Gene editing itself, it's not a silver bullet, it's not a fix-it-all or selection-all mechanism. EBVs and indexes, we really need to monitor overall genetic merit of animals and have balanced selection across all traits, where the benefit really of a precision tool like gene editing is that we can introduce a single high-impact trait or a couple of traits into a herd, but we really need EBVs, indexes, and gene editing if they work in tandem. That's where you get the potential for the most benefit. So gene editing won't be replacing EBVs or indexes anytime soon.
Yeah, that makes sense. So it sounds like gene editing will kind of just be another tool in the kit for people to use should they need it or want it as well, which is really nice to hear. I think it's never a bad thing to have lots of tools. So we spoke a bit before at the beginning of the episode about how this kind of looks like on-farm, but do you see this technology actually being applied on-farm in the future? Yeah,
certainly to some capacity being applied on-farm, because as I said earlier, that it can be applied anywhere IVF can be completed. So definitely in a small mobile lab on the back of a trailer, or in a hybrid model where part of the work's done on-farm, like the embryo collection, they're taken back gene edited in the laboratory and then come back for embryo transfer on farm again. So half and half. But yeah, and then in the next decade, I would expect that with the advances we're seeing in livestock IVF techniques and also CRISPR technologies, that it's possible for farmers, producers to get a small team, probably involves a vet, an embryologist and a molecular biologist, to come on farm if they want to introduce a particular trait into their herd. And this would be done like a routine reproductive service, similar to how embryo transfer or IVF is currently completed on farm. So in short, yes, I do see it partly being done on farm, either by a mobile lab type service or a hybrid based model.
That definitely sounds achievable, and it sounds like it would be a great option for people to be able to utilise as well. Are there any kind of risks in gene editing being misunderstood or misused at all?
Yeah, so gene editing is a pretty powerful tool, and with most powerful tools there is risk if it's not used responsibly. Fortunate we've got quite good regulation, both in Australia and in most places around the world, and also good science. We've got many decades of science backing the technology. Because without that, there's potential where we could be producing animals with traits that aren't actually beneficial to their local environment or the production system that they're in. For example, you've got myostatin-edited animals, those with extreme muscling traits. If they're put into a low-input, extensive system, they could be pretty vulnerable to heat stress or poor nutrition or disease. So we really have to ensure we've got good regulations. So what we're doing is ethical, and what we're doing is always benefiting farmers, benefiting the animals' well-being, and also a benefit to the wider community. I think overall, I hope I've given you some insight that I think that the benefits of gene editing overall far outweigh the potential risks for the technology in my perspective, my opinion.
Yeah, absolutely. You've given a great summary of that throughout the episode, so thank you. And everything's been super crystal clear, which is it's great, especially for gene editing, which seems to be quite new in Australia, and potentially there'd been some confusion around it with some other topics. So with gene editing being kind of new and not quite on the shelf yet in Australia, how do you see us bringing consumers along on the gene editing journey?
Yeah, it's super important that we do that, because we just don't want to be working in an ivory tower and already just come out with gene-edited animals that we have at our disposal. We've got to be transparent about what we're doing. So get out as scientists, educate people, and we've also got to be really open about the purpose of gene editing. We're not doing gene editing to be unnatural. Some people call it playing God. It's about using a safe and precise tool that we can deploy to try and improve animal welfare, improve the performance of animals, and also their disease resilience. And if we can achieve those three things, we have the ability to produce food and both fibre more sustainably, and it's pretty hard to argue against that. But I'm not naive. I get that there will be some opposition in the public, just as there is opposition to livestock farming in general. So we need to be respectful to everybody's opinion, but then listen to the consensus of opinion. And I think public perception really hasn't benefited from those first-generation GMOs, where we're making transgenic GMOs. Hasn't given us a head start in winning over public perception and consumers. So it's really important that we educate people, like I said earlier, on the differences between gene editing and traditional transgenic GMOs. Because gene editing has a range of benefits it can offer. We can deploy it safely. And if we can do that, it's pretty hard not to be on board with the benefits that it can potentially offer.
Yeah, no, that sounds good. Thanks for that, Gus. I think there's some really strong messaging in there, particularly around being transparent, and also education is definitely an important one. So just to kind of circle back and wrap up this whole episode, what do you see would be the most realistic use of gene editing in the next 5 years in some of the Australian herds or flocks, or I guess whatever sort of species we're looking at?
Well, that's a cool question to try and look into my crystal ball. In the next 5 years, I would think that we will see polled-ness being introduced into some of our horned breeds, so like full-blood Wagyu, through gene editing, and slick coat phenotype into some of the European breeds of cattle, because they're the two main traits that have been, I guess, most extensively researched and deployed in other countries around the world, so it's easy to see those being introduced into Australia as well. In sheep, I think we'll see shorter-tailed merino using gene editing, and potentially better breech traits as well. So we're reducing the frequency that mulesing and tail docking needs to be deployed. And I think these edits really align with what consumers want to see, or what the public expects from agriculture or animal agriculture. Gives animals a better life, and it's also easier for farmers to produce them because they've got less interventions to make. If I was to be optimistic and stretch that out to a 10-year time frame, I really hope that we have a better understanding of the genetics behind more complex traits, so things like methane emission. We know there's heritability there. So if we understand the genetics more, and we have CRISPR tools capable of improving those complex traits, we might be able to use those to transfer heritable methane emissions from breeds to breeds, and even other complex traits like feed efficiency, and that would be a great place to be in.
Thanks for sharing your crystal ball thoughts, Gus. They all sound very useful, and I'm sure a lot of people would find them quite valuable for their own enterprises as well. So I'd like to just say a huge thank you for sitting down with me today to discuss a lot of these really interesting concepts, and also just talking a lot about gene editing, which can be quite confusing for a lot of people, or even just a new topic. I think it's always really great, and circling back to what you said before about education, it's always great to put out some education that's very clear and concise, and I think that's exactly what you've delivered today with what can be a very complex topic from what I understand. So thank you so much for sitting down with me. I've really enjoyed this chat, and I've definitely learnt a lot, and I'm sure a lot of people listening have as well.
Yeah, thanks, Emily. It's great being on, and I hope I could offer some of your listeners and yourself a little bit of the insight into the gene editing and its application in livestock.
You definitely have done that. Thanks so much, Gus.
All the best. Bye.
Thank you. And that brings us to the end of this episode of Genetics in the Paddock with Emily. I hope you found our discussion as enlightening as I did. This episode was produced by the extensive livestock genetics team within the New South Wales Government. If you enjoyed this episode, please consider subscribing to our podcast on your favourite platform, leaving us a review and sharing it with your friends and colleagues. Your feedback and support help us grow and reach more people who are passionate about livestock genetics. If you have any questions, comments or suggestions for future topics or people you'd like to hear on the show, please feel free to reach out to me on emily.johnston@dpi.newsouthwales.gov.au. Thanks again for listening, and until next time.